name is Paula Alves. I'm currently the CEO of iBET, a private not-for-profit institute in Portugal. And I'm currently also the chairperson of ESACT, the European Society for Animal Cell Technology, and professor at the New University of Lisbon. ESACT is a society that is now 43 years old, and the mission is to promote animal cell technology and bring together scientists and developing equipment, developing biologics, and especially to motivate young people to come into this field that is so important for the development of biologics and biopharmaceuticals. In this meeting, I'm very happy to see that new things are arising and how demanding it will be for us as animal cell technologists and scientists working in this area the next 10 years, because really we are developing new ways of doing therapy, and cell therapy and gene therapy will be highly demanding on these technologies we have and we can provide to make these products in clinics. So for me it was very challenging to put together sessions on these topics, because these are not traditional topics on animal cell technology. So they were like seminal invited talks in the previous meetings, and here we have full sessions very well attended in these topics. So it's really nice for us as organizers of the meeting to see that this field is moving and that more and more attention is done to these fields of new therapies. I always worked in bioprocess development to produce biologics and characterize biologics. But from a biology perspective and biochemistry perspective. What I like is to combine the know-how I have in engineering with the cell biology, and as the cell is the host and for production of this type of products, what I like to do is exactly to think about in the cell point of view and how we can improve the cell health in a way by manipulating the metabolism to increase product quantity but also product quality. And now more recently, the cell per se is the product also for cell therapies. So again, by knowing more about the cell, how they communicate to each other, it's really important, and this is my drive in my research, is how to understand the cell behavior, the cell metabolism, how to characterize these products, how to have new tools to really have very well characterized products to have safer products in the clinics. To have a molecular way of treating the patients, not a disease type of approach like we have now, but looking at the patient and thinking about personalized medicines that are developed for that patient to address this disease in this patient. So this is my dream. At the meeting, we already had really challenged talks in the first and the second. That I don't want to name any talk in particular, but I was really excited about the talks that we have from invited speakers, keynotes, and several participants that were contributing to the program, the scientific program, showing, for instance, how we can, using synthetic biology tools, think of the cell as a circuit that we can manipulate and really shape the future of the cell, the direction of the cell, by manipulating the genes. This is wonderful. I think it's really exciting nowadays to see how this is moving. A lot of unknowns still, a lot to do, hopefully, always, but we are moving, and this is very rewarding.